The PCR is an enzymatic in vitro technique used to amplify exponentially a determinate DNA region located between two DNA regions, whose sequence is known for a complex nucleic acid mixture. For it is necessary three principal points. Number one, the mole from which the copies were generated. Second, the oligonucleotides, bots or primers specific to complementary ones to a portion of copies. And in number three, an enzyme able to synthesize the DNA using like the mole DNA. It's a DNA polymerase. The PCR reaction are necessary three principal steps. The number one is standard separation or denaturación. The second one is an hybridization of two oligonucleotide primers. And the third one is the DNA synthesis or extension. All three steps above are considered as a one cycle. It makes about 30 cycles in order to amplify the DNA template. Three different types of PCR. The first one is the colony PCR. Second one is a polymerase cycle assembly. And the third one is a droplet digital PCR. We are doing this experiment to run a successful PCR. We also want to show all the DNA amplification in an electrophoresis gel.
To perform the PCR, three reactions are prepared: the sample, positive control, and negative control. In order to make the three reactions, a mix is prepared. The mix consists in DNTPs, buffer PCR, water, primer forward and reverse, and DNA template. The final volume is divided by 3, so each tube will have the same amount of mix. In the test sample, 9.05 microliters of DNA and 40.95 microliters of mix are added. In the positive control, 1 microliter of PCR product, 8.05 microliters of water, and 40.95 microliters of mix are added. And in the negative control, 9.05 microliters of water and 40.95 microliters of mix are added. After preparing the three samples, they are taken into a thermocycler with the following conditions: the naturalization of the DNA at high temperature, 94 degrees for 5 minutes, elevation of the correct temperature, 56 degrees for 30 seconds, 68 degrees for 5 minutes, so that the DNA polymerase incorporates nucleotides complementary to the chains. The line 1.2a test sample shows three bands. The first one has a size of 495 base pairs and seems well defined. The following two has a size below the 100 pair base and seems distorted. The line 1.2b positive control shows two bands. The first one shows a sample excellent PCR product, and the second one has a size below 100 per base and seems distorted. The line 1.2c negative control shows three bands. The size of the bands are below 100 per base. The first one seems well defined and the second one seems distorted. In the results, in all the lines, multiple bands were obtained. In the electrophoresis, ideally, each line has to show one strong and well-defined band. The multiple lines in the electrophoresis indicate multiple primer sites for the primer within the target DNA. The multiple bands may be eliminated by raising the hybridization temperature, and adjusting the concentration of magnesium may be useful to eliminate unwanted products. Some bands were below 100 base pairs. This indicates primer dimers. To solve the problem, an adjustment in the concentration of magnesium may help to reduce primer dimers, but the redesign of the primer may be required to eliminate them completely.
The PCR is a very sensitive technique. Therefore, some conditions have to be taken in order to perform the technique. The PCR reaction starts to generate copies of the target sequence exponentially. Only during the exponential phase of the PCR reaction it's possible to extrapolate back to determine the starting quantity of the target sequence contained in the sample. In the result, in all the lines, Multiple bonds were obtained in the electrophoresis. Ideally, each line has to show one strong and well-defined band. The multiple lines in the electrophoresis indicate multiple primer sites for the primer within the target DNA. The multiple bands may be eliminated by raising the hybridization temperature